Assalamualaikum warahmatullahi wabarakatuh. My name is Fatin binti Mahadi from Faculty of Applied Science, and today I will be presenting about my final year project titled The Effect of Electron Withdrawing Group on SCHIFF-based ligands and copper and zinc metal complexes as antibacterial agent. This study and research was supervised by Professor Madya Dr. Karimah binti Kasim. For introduction, the research background in this study is about SCHIFF-based ligand. SCHIFF-based ligand is formed through the condensation reaction between an aromatic imine such as primary amine with aldehyde or ketone that produce an imine which contains functional group C double bond N. The presence of R or alkyl or phenyl group make the ligand a stable imine. An article from Soda Jose in 2018 mentioned that the SCHIFF base form through a reversible reaction and usually take place under acid or base catalyst or with the help of heat. The figure in this slide shows the general structure of SCHIFF base ligand. Article from Sakumari in 2021 mentioned that SCHIFF base play an important role in coordination chemistry. It able to increase the activity of drugs when they react with metal complexes. An article from Kara and Yaldu in 2019 mentioned that an electron withdrawing group is a substituent or a functional group within a molecule that has a tendency to pull an increase in electron density. There are three problem statements to be focused in the studies. First, a development of new drug is very challenging in biochemistry industry as it requires lots of discovery and synthesis to produce a new product which is safe to human cell and affordable to apply in medical industry. Second, SCHIFF-based ligand attract the medical field as it is a coordination structure that play an important role in possessing broad spectrum of biology activity. Third, these developments are able to achieve by using SCHIFF-base because these ligands contain the imine group that form from a reaction of carbonyl. There are three objectives in this study. First, to synthesize SCHIFF-based ligands and copper and zinc metal complexes. Second, to characterize SCHIFF-based ligands and metal complexes by using CHNS, elemental analyzer, melting point, FTIR spectroscopy, NMR spectroscopy, and computational analysis. Third, to study the antibacterial activity of SCHIFF-based ligands with their metal complexes by using this diffusion method. For literature review, there are four main focus in the studies by reviewing on selected articles that related to my studies. Here is the journal I use for synthesis method. Next, this journal I use for characterization for NMR spectroscopy. Next, for FTIR characterization, it was based on this journal. Lastly, here is the finding from the journal I use for antibacterial study. For methodology, here is the four main crucial part in the studies. First, the synthesis method for synthesis ligand that was used was conventional method in 2:1 ratio. Next, for synthesis metal complexes, the direct method was used in 2:1 ratio. Third, for the characterization, CHNS elemental analyzer, NMR spectroscopy by using proton and carbon analysis, FTIR spectroscopy, and lastly, computational analysis. And for antibacterial studies, the ligand and metal complexes was tested against E. coli, which is gram-negative, and S. aureus, which is gram-positive. Here is the synthesis of SCHIFF-based ligand 1 and ligand 2 with the proper structure of both ligands. Next, here is the synthesis of metal complexes for copper and zinc with the proper structure of the metal complexes. Next, move to the flowchart of the characterization for ligands and metal complexes. And lastly, here is the flowchart for antibacterial studies by using this diffusion method. From the data tabulated in this slide, it shows that the theoretical and actual percentage of carbon, hydrogen, and nitrogen composition was almost similar for most of the proper structure. Next, the complexation of zinc to acetate dihydrate with ligand 1 produced a lower percentage yield compared to others' complexation. This may be due to incomplete reaction where the ligands do not fully react with the reactants to form the products. The metal complexes exhibit high melting points compared to the free ligands as inferred from melting point data. This is likely to attribute to the enlarged size of the metal complexes in contrast to the ligands. The melting point for ligand 1 series show large range of difference compared to L2 series. The difference indicate the presence of electron withdrawing group, which is chlorine, in the compounds where it influences the temperature of melting point due to the strong polarization effect. Next, for characterization using proton and mass spectroscopy, the presence of aromatic proton C double bond N for ligand 1 appears at 8.16 ppm as singlet peak. This peak indicates the conformation of SCHIFF base that presents in the ligand. The single peak of C double bond N appears in the spectra, indicating the structure of the ligand is symmetrical as it appears on one side of the structure. The peak for aromatic hydrogen appears in multiplet at the range of 7.16 to 7.57 ppm. For ligand 2, the presence of aromatic proton C double bond N for ligand 2 appear at 8.15 ppm as singlet peak. This peak indicates the conformation of SCHIFF base present in the ligand. The single peak of C double bond N appears in the spectra, indicating the structure of the ligand is symmetrical as it appears on one side of the structure. The peak for aromatic hydrogen appears in multiplets at the range of 7.16 to 7.63 ppm for ligand 2. From the table of 13 carbon NMR data for ligand 1, the resonant peak for aromatic carbon C double bond N for ligand 1 was observed in the downfield region at 153.80 ppm. The chemical shift of C double bond N for ligand 1 was slightly distributed due to the presence of substituent of electron withdrawing group, which is chlorine, in the compound. The signal peak for CCl in ligand 1 was observed at 151.74 ppm. The aromatic carbon for ligand 1 appears at the range 129.22 ppm to 151.74 ppm. From the table of the 10C NMR data for ligand 2, the resonant peak of aromatic carbon C double bond N for ligand 2 was observed in the downfield region at 151.77 ppm. The aromatic carbon for ligand 2 appears at the range of 126.86 to 143.95 ppm. The characterization of FTIR spectroscopy for ligand 1 and ligand 2 series, the presence of azomatine C double bond N indicate the presence of SCHIFF base in the ligand and metal complexes. The trend for vibrational peak of the azomatine in metal complexes for the ligand 1 and ligand 2 series were shifted to lower frequencies compared to their parent ligand peak. This shifting indicates the coordination between the azomatine nitrogen and metal center that had taken place. Next, for computational analysis, the energy band gap of ligand 1 is 1.368, while for ligand 2 is 4.294. For ligand 1, the energy band gap is lower compared to ligand 2 due to the presence of electron withdrawing group in the compound. The electron withdrawing group affect the electronic structure and energy level of the compounds, which make it easier for the molecule to undergo electronic transition. Then, the electron withdrawing group tend to reduce electronic density in certain region. From the table, it shows that the antibacterial activity of ligand 2 and its metal only show positive result again gram-negative, which is E. coli, for both concentration. The negative result for L2 and its metal against the gram-positive may be related to the concentration and sensitivity of SCHIFF base against the bacterial agent. The differences of inhibition zone tested against E. coli and S. aureus when testing ligands and metal complexes against both bacteria was attributed to the distinct cell wall structure of this bacteria. The thicker peptidoglycan layer in the gram-positive of S. aureus made it more susceptible to interact with ligand and metal complexes, resulting in inhibition zone. Thus, to conclude, the antibacterial activity of ligands and metal are better generally in 100% concentration of DMSO compared to 10% concentrations of DMSO. From the finding, it shows that the metal complexes inhibit better antibacterial agent compared to the ligand. The significant importance of these compounds owing to their unique chelating properties due to the presence of essential nitrogen donor atom. In conclusion, the percentage heat obtained from the synthesis was at range 19.51% to 76.45%. In the IR spectra, the shifting of azomatine aromatic ring indicate the presence of coordination metal ions with imine nitrogen and benzene ring. In NMR spectra, the chemical shift of imine group which confirm the presence of azomatine bond. The antibacterial studies show that the ligand and metal complexes are better antibacterial agent in E. coli compared to S. aureus in both concentration. There are 4 recommendations that can be improved in the studies. First, the reflux time for the ligands and the metal complexes can be lengthened to ensure that the reactions can be fully react. Second, the sample must be in suitable concentration before it was characterized by NMR spectroscopy. Third, use X-ray diffraction for characterization. Fourth, for antibacterial activity, the screening of inhibition zone can use another gram-positive bacterium such as Streptococcus mutans. That's all from me. Thank you.